Welcome to today's zoology class. So in the last few classes, we have studied the monohybrid cross of governmental and two principles deducted as a result of this monohybrid cross, law of dominance and law of segregation, and also certain deviations like incomplete dominance, codominance, multiple allelism and pleiotropy from Mendel's findings, revealed later by certain other scientists. So anyway, so far we have studied the details of monohybrid cross. So we remember that monohybrid cross was a hybridization experiment in which only a single character was considered. We have seen the example of a hybridization between tall and dwarf. So the character was height. Only a single character was considered in those experiments. But Gregor Mendel wanted to study the influence of both two characters, in the process of inheritance. Or he wanted to know how the process of inheritance of one character influences the process of inheritance of other character. Or how two characters at a time are inherited to the next generation. So for this, he decided to conduct hybridization experiments where two characters are considered at a time. So in the monohybrid cross, he considered only one character at a time. Or he conducted hybridization experiments by using a pair of contrasting features, contrasting traits of a single character. Later he wanted to experiment, or later he wanted to conduct hybridization experiments by using two characters at a time, by considering two characters at a time, or two pairs of contrasting traits at a time. So, this hybridization experiment, this cross, where two characters are considered or two pairs of contrasting features, contrasting traits are considered at a time, is called dihybrid cross. Di means two. So, two characters are considered at a time. This is called dihybrid cross. Now, in this class, we will be discussing about this dihybrid cross and related aspects. Okay, straight away we will go to the a classical experiment that Gregor Mendel conducted. So here the characters selected for hybridization experiment were shape of the seed and the color of the seed. So shape of the seed is one character and color of the seed is another character. When we examine the chart provided to you in the previous classes, showing the dominant characters and recessive characters, you must be remembering that. In the chart, as you can see here also, not the shape of the seed. As far as the shape of the seed is concerned, two contrasting traits are there. Round seeded pea plants are there and wrinkled seeded pea plants are there. From the chart you can understand that roundness or round character, round trait is dominant over wrinkled trait. So the round seed is the phenotype, is the dominant phenotype, and wrinkled phenotype is the recessive phenotype. From the chart you can understand that. Then color of the seed. As far as the color of the seed is concerned, there are two contrasting traits. Yellow seeded pea plants are there and green seeded pea plants are there. Yellow seed is dominant over green seed. So Gregor Mendel selected these two characters, the shape of the seed and color of the seed, for his dihybrid cross. Or two pairs of contrasting features, contrasting traits he selected. Round seeded pea plants and wrinkled seeded pea plants, or a pea plant with a round seed and yellow color crossed with a pea plant with a wrinkled seed and green color. So these are the parents for hybridization experiments. Round seeded, a pea plant with a round seed and yellow color crossed with another pea plant with wrinkled seed and green color. Okay. Now let us write down the genotype of these parents also. So according to the Mendel's factor hypothesis, every character is controlled by a pair of factors, a pair of alleles. So the round shape of the seed is represented by or is produced by a pair of alleles. Here it is represented as capital R because round shape is dominant over wrinkled shape. So we denote it with the capital letters. Capital R. Yellow color is also dominant over green color, and that is represented by capital Y. So capital R for round shape, capital Y for yellow color. Then wrinkled seed is represented by wrinkled phenotype is recessive over round. So wrinkled phenotype is represented by small r, and green color, which is recessive, represented by small y. Okay, we are representing the recessive character with a small letter of the dominant factor. So capital R, capital Y is crossed with small r, and small y. So this is a dihybrid cross. Now let us come to the formation of gametes. Let us see how gametes are formed here. Remember, these parents are homozygous. So they produce all the gametes with the same genotype, same allelic combination. Every homozygous individual, a plant for homozygous for a particular trait, will produce the gametes with all the gametes with the same alleles. So as they are homozygous, they produce same gametes. All the gametes will be similar, same. So what will be the allelic combination or factors in the gametes? Here we should consider there is a difference here from the monohybrid cross. We should remember that. Again, let us go to the law of segregation. According to law of segregation, during gametogenesis, the pair of factors separate and enter into separate gametes. So that is applicable here also. So alleles for the shape roundness are capital R, capital R. So during gametogenesis they are separate. One R enters into one gamete and the other one enters into another gamete. So they are separated. In the same way, capital Y representing the yellow color also segregate. So every pair of factors representing each and every character separate, segregate during gametogenesis. So capital Y also separate. But here you should understand that a gamete is the unit of an organism. A gamete is the unit of every organism, every sexually reproducing organism. Sperm is the unit of a male individual. Ovum is the unit of female individual. As these are units, these gametes are units, they should contain alleles for every phenotypic expression. Okay, I hope you understand. The gamete should contain the allele for, at least a single allele for every phenotypic expression. So, when gametes are produced, it should contain an allele for shape and an allele for color. So according to law of segregation, the alleles are separated and the gamete should contain one allele for round shape and one allele for yellow color. Okay. So we get alleles with capital R and a capital Y. This is the gamete. Tall and dwarf in the crossലേക്ക് നമുക്കൊന്ന് പോയാൽ നമുക്ക് മനസ്സിലാവും അവിടെ gamete ഉണ്ടായപ്പോൾ അവിടെ ഇത് തന്നെയല്ലേ സംഭവിച്ചത്. അവിടെ ഒരു ക്യാരക്ടർ മാത്രമാണ് നമ്മൾ കൺസിഡർ ചെയ്തത്. അതുകൊണ്ട് ടോൾനെസിന്റെ ഒരു അലീൽ ഗമീറ്റിലേക്ക് വന്നു, ഡ്വാർഫ്നെസിന്റെ ഒരു അലീൽ ആ ഗമീറ്റിലേക്ക് വന്നു. ഇവിടെ രണ്ട് ക്യാരക്ടർ ഉണ്ടല്ലോ, 2 characters ആണ്. അപ്പോൾ ഓരോ ക്യാരക്ടറിന്റെയും ഓരോ അലീൽ വെച്ച് ഗമീറ്റിൽ വരണം. അപ്പോൾ ഷേപ്പിന്റെ ഒരു അലീൽ ഗമീറ്റ് ഉണ്ടാവണം, കളറിന്റെ അലീലും ഗമീറ്റ് ഉണ്ടാവണം. So as far as a round yellow plant is considered, its gamete should contain one allele for roundness, round character, round shape, and another allele, an allele for yellow color is also should be there in the gamete. So the gamete should contain an allele for round trait, roundness, and one allele for yellow color. So the gamete is capital R, capital Y. And what about the other parent? Here also the same. Suppose the recessive plant is the female and when ova are produced, this ova should contain one allele for the shape. Here it is wrinkled shape, one allele for shape and one allele for color. So the gamete should contain capital small r, small y. So in this way, the gametes are formed. So these gametes fertilize, new generation, F1 generation is formed, and the F1 Gregor-Mendel noticed that all the plants produced were round yellow. We know that this should be the case because we have already seen in the monohybrid case, the case is the same. This is according to the law of dominance. According to the law of dominance, the dominant character appears in the F1 and the recessive character is masked or hidden. That is what is happened here also. The dominant characters are round and yellow. Both are dominant characters. These characters appeared in the F1. So all the F1 progeny are round, yellow, but notice that they are heterozygous. What is the genotype? Capital R, small r, capital Y, small y. As a dominant allele for roundness is there, the plant produces round seeds. As a dominant allele for yellow color is there, the seeds are yellow in color. So the F1 progeny are all with round seed and yellow color. They are heterozygous. Now, as in the case of monohybrid cross, Mendel here also proceeded to form the next generation, F2 generation. So, what did he do to produce the F2 generation in the case of monohybrid cross? He self-pollinated the F1 progeny. Mendel self-pollinated the F1 progeny. So here also he conducted the same thing. He did the same thing. He self-pollinated the F1 progeny. And he produced the F2. In the case of F1, so in the case of monohybrid cross, the F2 produced the progeny in the ratio 3:1. And we called it the monohybrid ratio. When 3 tall plants were produced, for every 3 tall plants produced, 1 dwarf plant was produced. That is the ratio between tall and dwarf. But here, in the case of dihybrid cross, the F2 progeny were produced in the ratio 9:3:3:1. He got a ratio, a phenotypic ratio, that is the phenotypic ratio. He got a phenotypic ratio 9:3:3:1. Let us examine what this ratio stands for. 9 represents yellow round. 1 represents wrinkled green. This is okay, this we can understand. Because this was what produced in the monohybrid cross also. When 3:1 ratio, we obtained 3:1 ratio, 3 plants were tall and 1 plant was dwarf. Just like the grandparents. Just like their grandparents. The grandparents were tall and dwarf. So 3 plants were tall and 1 plant was dwarf. So when the dihybrid cross, when he got 9:3:3:1 ratio, 9 plants were round yellow, just like one parent, and 1 plant was just like the other parent, green, wrinkled green. So just like the grandparents, they got the F2 progeny. But apart from the parental combination, this we call the parental combination, resembling the parents. So the 9 plants, out of the total plants produced, 9 part of the total progeny were just like one parent. This is the parental combination, just like one grandparent, of course. This we call the parental combination, round yellow. And the 1 plant is like another parent. There is also a parental combination. But what about the other plants? 3 and 3. 3 part of the total progeny were round green. And the other 3 part of the progeny were wrinkled yellow. This is not like the parents. This is the difference from the parental combination. There were no parents like that. There were no parents like this. So, this we can call new combination or recombination. They are called recombinants. Recombinants. So, this was the progeny produced in the ratio 9:3:3:1 in the case of F2 of a dihybrid cross. So, Gregor Mendel tried to explain how this happened. How this 9:3:3:1 or mainly how this recombination occurred. That was his main target. How this recombination occurred. Parallel combination we know, we already explained that in the monohybrid cross. That is possible. And we already studied how does it appear. There is no confusion with that. But how does the new combination appear? That needs some explanation. And based on, by using his logic and analytical capacity, analytical ability, he explained it through his third law of inheritance. So here comes the third law of inheritance or third principle of inheritance put forward by Gregor Mendel, that is called law of independent assortment. What is that? Law of independent assortment. Law of independent assortment. What do you mean by assortment? Assortment means വ്യത്യസ്ത ഘടകങ്ങൾ തമ്മിൽ ഒന്നിച്ചു ചേരുക, മിക്സ് ചെയ്യപ്പെടുക എന്നൊക്കെയാണ് അസോർട്ടിന്റെ അർത്ഥം. അത് പല ഘടകങ്ങൾ, പല ഫാക്ടേഴ്സ് തന്നാൽ വ്യത്യസ്ത ഫാക്ടേഴ്സ് പരസ്പരം മിങ്കിൾ ചെയ്യുക. ഇതിനാണ് നമ്മൾ അസോർട്ട് ചെയ്യുക. അപ്പോൾ നമുക്ക് എന്താണ് ഈ ലോ ഓഫ് ഇൻഡിപെൻഡന്റ് അസോർട്ട്മെന്റ് എന്ന് നമുക്ക് നോക്കാം. അപ്പോൾ അദ്ദേഹം പറഞ്ഞു, ഡ്യൂറിംഗ് ഗമീറ്റോജെനസിസ് of the F1 progeny, alleles representing different characters or different traits assort independently and form gametes. So let us see how it happens. So let us see the genotype of the F1 progeny. Capital R, small r, capital Y, small y. So this is the genotype of the F1 progeny. F1. So according to law of segregation, the pair of factors separate. That's already there. They're sure. For certain they segregate. So the capital R, small r, they segregate, separate. Capital Y, small y also separate, segregate. These are alleles representing a particular character. Capital R, small r represent the shape of the seed. Capital Y, small y represent the color of the seed. So the pair of factors separate. After separating or after segregating, they assort independently. That is what here Mendel taught. Assort independently. Or without any prejudice, without any predetermination, they are combined with the alleles of the other character. They combine with the alleles of the other character. Let us see how these alleles can be combined. Let us take the case of capital R. It is already segregated. Now it is free. Capital and small r are free. They are segregated. So after segregation, the capital R can be assorted in different ways. with the other allele, with the alleles of the other character. That is the color. Capital R is the allele of the shape. And according to Grigor-Mendel, it can assort with the alleles of the color in various ways, independently. So let us see how it assorts. Capital R can be assorted with capital Y. Can go with capital Y. So we'll get a gamete, capital R, capital Y. You can also assort with a small y. So capital R, small y, will get another gamete, capital R, small y. Now let us come to the small r, the recessive allele. You can also assort with the, it's the allele of the shape. You can also assort with the color alleles for the color. So one possibility is it can assort with the capital Y. It can go along with the capital Y. So we will get a gamete with a small r, capital Y. It can also go with or assorted with the recessive allele for color, that is small y. So we will get a small r, small y, small r, small y. So how many gametes is possible now? 4 gametes are possible. We are talking about possibility, okay? So 4 different types of gametes are possible here. Which are they? One is, so while constructing the gametes here, we should understand, we should remember that the gamete should contain one allele of every character. Here only 2 characters are there, shape and color. So the gamete should contain one allele for the shape, one allele for the color. Okay. So in that way, we have 4 gametes here. One is capital R, capital Y. So the shape allele is there, color allele is also there. One gamete is small r, capital Y. Small r, capital Y. There also shape allele is there, color allele is there. Another possibility is small r, capital Y, and another possibility is small r, small y. So which are the alleles here, which are the gametes here? Once again let us see. Capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. So in this way, 4 different gametes can be formed. So these 4 different gametes are formed according to Grigor-Mendel due to the process of independent assortment. After the alleles are segregated, they assort independently. That means they combine with or they go along with the alleles of the other character independently, freely. So in that way, we get 4 different gametes. So whatever the other plant, the recessive, whatever the other plant, the same. Because there is self-pollination. F1 is self-pollinated to get the F2. So one plant produces these 4 gametes. Suppose these are male gametes. And the other plant also produces the same gametes, 4 different types of gametes, which we can consider as the female gametes. Okay, let us draw the Punnett square here, as you can see here, the Punnett square, and place the gametes, male gametes and female gametes, and see the progeny. As you can see here, this Punnett square, total 16 progeny produced. 16 progeny are depicted here in this Punnett square, and they are in the ratio 9:3:3:1. You can see here. How many types of plants are there? 9 plants are round yellow. 3 plants are round green, and 3 plants are wrinkled yellow, and 1 plant is wrinkled green. So we get 9:3:3:1 ratio, and this ratio is called the dihybrid ratio. So dihybrid ratio. By observing this Punnett square, you see the genotype also. All these 9 round yellow plants do not have the same genotype. Okay, they all have different genotypes. One is capital R, capital R, capital Y, capital Y. Another one may be capital R, small r, capital Y, small y. Another may be capital R, capital R, capital Y, small y. So in that way you can you observe that and understand the genotype also. So this ratio is called the dihybrid ratio. So by conducting this experiment, he studied how the one character influences the inheritance of the other character. And he put forward the principle, the third law of inheritance, that is law of independent assortment. Okay? So we will wind up the class here. We will see you in the next class. Bye.